NQO1. NQO1 is called NADPH dehydrogenase quinone 1. NQO1 serves as a quinone reductase in connection with conjunction reactions of hydroquinones involved in detoxification pathways, as well as biosynthetic processes such as the vitamin K-dependent gamma carboxylation of glutamate residues and prothrombin synthesis. The catalytic activity for NQO1 is NADPH plus a quinone yields NADP plus a hydroquinone. The cofactor for this gene is FAD. NQO1 is inhibited by dicumarol. Dicumarol is a naturally occurring anticoagulant that functions as a functional vitamin K depleter, similar to warfarin, a drug that dicumarol inspired. It is also used in biochemical experiments as an inhibitor of reductases. The molecular functions for NQO1 are cytochrome B5 reductase activity acting on NADPH, identical protein binding, NADPH dehydrogenase quinone activity, poly-A RNA binding, superoxide dismutase activity. The biological processes are aging, cellular nitrogen compound metabolic process, negative regulation of catalytic activity, nitric oxide biosynthetic process, positive regulation of neuron apoptotic process, regulation of cellular amino acid metabolic process, response to estradiol, response to ethanol, response to nutrient, response to toxic substance, small molecule metabolic process, synaptic transmission, cholinergic, xenobiotic metabolic process. The enzyme NQO1 is a potential target for personalized medicine, as it is overexpressed in many solid tumors. In normal cells, NQO1 is inducibly expressed, and its major role is to detoxify quinones via bioreduction. However, certain quinones become more toxic after reduction by NQO1, and these compounds have potential as selective anti-cancer agents. Several quinones of this type have been reported, including mitomycin C, RH1, E09, streptonigrin, beta-lapicone, and deoxyniboquinone, also known as DNQ. This gene is a member of the NADPH dehydrogenase quinone family and encodes a cytoplasmic 2-electron reductase. This FAD binding protein forms homodimers and reduces quinones to hydroquinones. This protein's enzymatic activity prevents the 1-electron reduction of quinones that results in the production of radical species. Mutations in this gene have been associated with tardive dyskinesis, also known as TD, an increased risk of hemotoxicity after exposure to benzene, and susceptibility to various forms of cancer. Altered expression of this protein has been seen in many tumors and is also associated with Alzheimer's disease, or AD. Alternate transcriptional splice variants encoding different isoforms have been characterized. We have developed a simple real-time cellular assay for NQO1 and show that PMET plays a significant role in DMNQ redox cycling via NQO1, leading to cellular toxicity in cells with high NQO1 levels. There could be an increased risk of hematotoxicity in individuals with the TT-NQO1 genotype. A protective role for NQO1 against benzene-derived quinones in bone marrow in situ required reconciling with the observation that freshly isolated bone marrow, mononuclear, and progenitor cells fail to express NQO1. NADPH quinone oxidoreductase NQO1 catalyzes the 2 or 4 electron reduction of numerous endogenous and environmental quinones. Example, the vitamin E alpha tocopherol quinone, menadione benzene quinones. Mutations have been associated with tardive dyskinesia, also known as TD, an increased risk of hemotoxicity after exposure to benzene, and susceptibility to various forms of cancer. The expression of NQO1 is increased in liver, colon, and breast tumors and non-small cell lung cancer, also known as NSCLC, compared with normal tissues. Moreover, expression levels are also elevated in developing tumors, suggesting a role for NQO1 in the prevention of tumor development. NADPH quinone oxidoreductase NQO1 detoxifies benzene-derived quinones via 2-electron reduction, preventing redox cycling and covalent modification of cellular components. CYP1B1 COMT with a combination of NQO1 expression can lead to estrogen dominance. Low NQO1 activity and increased formation of depurinating adducts resulting from unbalanced estrogen metabolism can be critical factors leading to the initiation of breast cancer. If you have any concerns about NQO1, contact a practitioner familiar with epigenetics and nutrigenomics.